Before I begin, I would like to thank the organizers and the reviewers for the honor of this award, and it's a pleasure to share our work with you today. With this presentation, I'll give an overview of our research that crosses between visualization and biomedical illustration techniques to address challenges in exploring, analyzing, and communicating aspects of human physiology. Human physiology encompasses the millions of interconnected biochemical processes that are integral to keeping us alive. And human physiology is complicated, incorporating elements of all basic sciences as well as more specialized fields such as systems biology and medicine. Processes in physiology span several scales of space and time that make it a truly multidisciplinary field of study. For instance, a single structural modification of a molecule could require a structural biologist perspective, but the cascading effects of this change that can manifest in tumor metastasis requires a different set of expertise to identify and treat. With this breadth of physiology, it's understandable that myriad data types exist to capture aspects of this science in order to help guide therapy, monitor disease progression, and to identify new diagnostic markers. And these data are only increasing, with technological advancements leading to higher resolution and new paradigms to acquire such data. Visualization can be a powerful means to help make sense of these data, with promise to help experts in key analysis challenges, such as identifying possible markers for disease, to understanding how variation scales from an individual to a cohort level, and to relate findings to the source data. Communicating these research findings to various stakeholders, however, is often managed through biomedical illustration. This concept, rather than data-focused field, can, for instance, fill gaps where data are unavailable and abstract information to a degree that's audience appropriate. Our research expands on the multidisciplinarity of physiology by exploring opportunities for visualization and biomedical illustration to discover and communicate information about physiology. This cross-disciplinary perspective is in part encouraged by current societal demands for compelling and understandable visual representations of physiology, which necessitate new methods that are spanning disciplines. More specifically, our research can be broken down in the following way. It's scoped to visualization for human physiology over 5 publications spanning the molecular to cohort scale, which contribute to the areas of theory, empirical findings, method, application, and research replicability. We focus on 2 actor groups that include experts in an aspect of physiology and non-experts who lack this topical expertise, and this can apply to the general public. And we divide our cases according to high-level visualization task, which we've adapted from Bramer and Munzner, in 2 areas: exploratory analysis for multifaceted data that are oriented to experts, and visual communication, which can be more general. Our focus on these cases arose from our state-of-the-art report in visualization for physiology as 2 key areas of opportunity for further research. Looking first at our work in exploratory analysis of multifaceted data, our efforts focus on the identification of biomarkers, meaning molecules that can indicate a disease, that can lead to early diagnosis and assessment of diseases and disorders of the central nervous system. We take 2 approaches in this space. The first is a data-specific design study to address the gap in visualization research for magnetic resonance spectroscopy, also known as MRS. MRS is a non-invasive technique used to estimate concentrations of certain biomolecules, known as metabolites, in living tissue that are often undetectable in other modalities. It has promise for use in tumor and radiotherapy studies, as well as in early detection of, for example, Alzheimer's disease. However, this modality is challenging to interpret, with limited clinical tools or visualization research that support translation from these acquired signals into clinically useful markers. We conducted a year-long collaborative study alongside cognitive neuroscientists, MR physicists, and MR engineers at the University of Bergen and Haukeland University Hospital in Norway to understand the challenges, tasks, and workflows for interpreting MRS data. From this, we developed a novel method to explore and compare ratios of metabolites at different levels of detail. We achieved this through a layered heat map matrix. The first layer, shown here, takes inputs that can constitute single or multiple voxels, which are the sample regions, that contain metabolites from one or many locations, individuals, time points, or task states, which can be active, where the individual is performing a task while in the scanner, or passive, which is no task while in the scanner. Each cell is the average metabolite concentration from the x-axis to the average metabolite concentration in the symmetrical position on the y-axis, with the value in the cell where they meet mapping to color. Within each cell of the heat map matrix, we nest a set of glyphs that break out these attributes of interest. A rectangle glyph indicates the voxel sample region. A circle indicates each individual sampled in this area, which nests inside, which is divided if the acquisition had a dual task state. And this expands to maximize pixel space with a sparkline for time series data. This method is integrated in the SpectraMosaic application, which provides a visual workflow that links patient, structural, and spectral information. In our evaluation with 3 experts, we found that SpectraMosaic enables rapid and iterative exploration of different permutations of metabolite ratios and how they vary across attributes. These different views of the data allow for quick and easy quality assurance of the data. For instance, to identify the hippocampal region here as the oddity in a study over different cortical tissue locations. Expanding beyond a specific data source, clinical investigation of biomarkers for early disease diagnosis often requires extensive analysis of multifaceted cohort data from multiple data sources. Analysis of these data can be overwhelming. The data are heterogeneous, high-dimensional, with many missing items, kind of like a gear room. While visual analysis approaches incorporating dimensionality reduction methods can make analysis more manageable, they risk losing subtle relationships that could be interesting to experts. A key motivation for our work came from an ongoing collaboration with colleagues at the University of Bergen and the MMIV, as well as researchers at Otto von Guericke University and clinical neurologists at the Center for Behavioral Brain Science in Magdeburg, Germany, who study cerebral small vessel disease. This is a common disorder of old age associated with abnormalities of small blood vessels in the brain that are responsible for 1 in 5 strokes worldwide. However, its etiology is largely unknown, and our colleagues needed a method beyond their standard analysis protocols. Our proposed solution, DimLift, can lift subtle patterns between dimensions for users to flexibly explore dimensional subspaces in a hierarchical manner to generate hypotheses early in the analysis process. The core idea of our approach is the algorithmic formation of dimensional bundles in a semi-automated, iterative fashion, as this diagram illustrates. Each bundle consists of dimensions that contribute similarly to the overall variance of the dataset. Users can explore and modify these bundles via a hierarchical parallel coordinates plot, where each parallel coordinates axis is a bundle, and glyphs above provide information on bundle characteristics. Interactions allow for rebundling, for exploration of contributing dimensions, and of relationships of items and dimensions between and within the bundles. With DimLift, we found that clinical researchers can augment their existing analysis pipeline to discover relationships that otherwise may take hours to uncover, if at all. For instance, observing the bundling of lacunes and microbleeds together as discovered in this session. These findings can then be interrogated in more targeted analytics tools. Research doesn't end with expert analysis, though. Experts' findings have to be shared with stakeholders in other domains with different expertise to carry findings effectively from the lab into policy. And it's natural to expect that experts in a topic could have different preferences and criteria to evaluate a visual communication relative to a non-expert audience. This impacts the success of a graphic in communicating a scenario. A goal of our first study in this communication space was to gain insights into how visualization and biomedical illustration techniques are used and assessed by different audience groups. Designed over a series of expert interviews and focus groups with professional biomedical illustrators and visualization researchers across Europe and North America, our interdisciplinary study with researchers from the University of Bergen, Otto von Guericke, and Johns Hopkins University focuses on common communication scenarios of 5 well-known physiological processes and their standard visual representations. We frame these scenarios in a survey with participants' self-rated expertise on the physiological process of interest spanning from minimal to expert knowledge of a given topic. Our results show many shared preferences between expert and non-expert audiences, although experts and non-experts use slightly different criteria to define ineffective visualizations. We also found that some illustrative conventions, for example glows, are ambiguous in meaning, and results suggest that participant background can influence preference. Our findings present numerous opportunities for the continued convergence of visualization and biomedical illustration techniques to visually describe physiology. Some of the aesthetics questions led to our next study for the value of soft guidelines and best practices for color and molecular visualizations to give more semantic meaning to visualization at this scale, which is incredibly abstract to the broader population and is increasingly important to understand with increased exposure to these structures in the public space, as we saw during COVID-19. Similar to how the CPK coloring scheme has given instant meaning to atoms, we consider if something similar could and should be done at the molecular scale. We suggest that guidelines for aesthetics can help those without artistic training to make more visually pleasing visualizations of their research, and that color can be used to leverage perceptual cues to make a scene easier to interpret, and to apply color to ensure that molecular visualizations are read correctly, for instance, coloring by functional or structural groups. To support open science, our application code is certified through the Graphic Stamp Replicability Initiative for SpectraMosaic and DimLift, and we created publicly available data packages for our empirical work. The myriad interconnected biochemical processes contributing to physiology are complex yet necessary to understanding life. Increasing volumes and types of data describing physiology are necessitating interdisciplinary teams to solve challenges in discovering and communicating important features of these data, with visualization and biomedical illustration both integral in allaying these challenges. Our body of work is enriched for its interdisciplinary, but it has its limitations. We couldn't cover the entire scope of physiology and instead rely on representative topics and gaps identified in our state-of-the-art report. Many of our studies are small and require larger evaluations with more diverse groups to confirm our preliminary findings. Lastly, our application approaches target clinical research, but an ultimate goal is to see our work realized in the clinic for more immediate and high impact. Visualization for physiology has a really bright future. New acquisition paradigms can benefit from new visualization methods, and processes spanning broad scales of space and time are an unsolved challenge to visualization, which is especially the case at the molecular scale. Experts often need to relate their insights back to the raw data, and continued research that links insight to source is important for expert trust and adoption. And this is where investigating different communication strategies can play a role in establishing credibility. And this credibility question is also important to study for non-expert audiences. I'll wrap up here by acknowledging our funding sources, and last but not least, to express my gratitude to all co-authors and collaborators who contributed to this body of research. This really would not have been possible without them. Thank you for your attention.